Hello everyone, welcome back to the channel Bio-Microbio-Logy. I am here again with a new topic, endospore staining, the most common microbiological experiment. In this video, we are going to discuss about various things of endospore staining, like what is endospore and its structures, example of endospore forming bacteria, types of spore, then principal applications and procedures of endospore staining, and how dyes act on endospore. Now, what is endospore? Endospore is the dormant-like structure that formed within a cell. When environmental condition is adverse in case of limited amount of nutrients, extreme heat, radiations, harmful chemicals, bacteria can't survive as because of the vegetative growth is stopped or ceased. A number of gram-positive bacterial genera such as Bacillus, Clostridium, Sporocercina and some other bacterial cells have the ability to convert themselves into a dormant-like structure called endospore, which helps them to survive in the harsh environmental conditions. Endospores can form within a bacterial cell or occurs as free spores. Here it's showing the structure of an endospore forming bacterial cell where endospore formed within the cell. The spore contain an outermost layer called exosporium attached with the spore coat. After that there is an outer membrane, then inner membrane and core of the spore is present. And in the core of the spore that contains genetic materials of the cell. Here it's notable that endospores can germinate back into vegetative cells, that's an active state of bacterial cell that undergoes metabolism when environmental conditions are favorable. Types of spores. As we have already mentioned that an endospore can formed within a cell when it can formed in central position that called central spore, when formed in terminal position that's called terminal spore, then subterminal spore, and there is a solen tip like structure of the spore called solensporangium. Some example of endospore forming bacteria. Some endospore forming bacteria such as Bacillus and Clostridium species. These bacterial genera naturally grow in soil but having significant role in some clinical implications by causing infections to human. Such as example, Clostridium tetani causes tetanus, Clostridium botulinum produces botulinum toxin that causes paralysis, Bacillus anthracis causes anthrax in cattle and human, Bacillus cereus causes food poisoning. Now we are coming to the main point, endospore staining. As in the last slide we discussed about the structure and the layers of the spores. That's how the structure of an endospore looks like, as it contains a tough outer layer that offers them a great resistant capability at any situation. In case of some pathogenic bacteria, it acts like virulence factor and not easily affected by any chemicals, disinfectants or heat. That's why to study their morphology, it's difficult to stain endospore forming bacteria with some basic dye that can't penetrate their layer. Hence, a special stain is applicable that contains a special dye along with heat stain processing, and the staining procedure is known as endospore staining. The principle of endospore staining. As the word endospore defines a spore-like structure present within a cell. The main purpose of the special staining procedure is to stain the spore of a vegetative cell to check whether the cell contains an endospore or the cell normally exists in a reproductive stage or freely exists in the sample. Staining helps us to see the cell and spore through microscope. The main principle of endospore staining is it's a differential staining which distinguishes the dormant spore from its vegetative part of cells or other vegetative cells. Applications of endospore staining. Some bacterial diseases such as tetanus, anthrax, gas gangrene, botulism and pseudomembranous colitis are generally caused by some species. Those can form endospore. The capability of forming resistant spore of the pathogenic bacteria already mentioned in previous slide made them virulent to themselves while infecting host. Endospore staining has great application in clinical diagnosis of identifying of the infections mentioned above. It also have application in microbiological and pharmaceutical research for the potential application and manufacturing of new drugs. So here the staining procedure. In 1922, Dornat first introduced endospore staining method where heating process was lengthy that resulted in a differential staining of the endospore and the vegetative cell in a same sample. In this process of endospore and free spores appeared in green in color while the vegetative cell retained the red color. In 1933, Sifu and Fulton modified the Dornat's endospore staining method and made it more faster. The Sifu-Fulton method is commonly used by microbiologists. The procedure is described next. The Seyfert-Fulton method of endospore staining. In the basic laboratory experiment, the simplest, also a differential endospore staining technique is the Seyfert-Fulton method and is followed in most laboratory protocols. The staining procedure is described below. First of all, the reagents are used: 1. Malachite green dye, the primary stain; 2. Distilled water, the decolorizing agent; and 3. Counter stain, safranin. And the equipments are needed here: glass slide, inoculating loop, Bunsen burner, water bath. At first, take a clean grease-free glass slide and put a drop of sterile water on it. Then carefully take a loopful culture of a bacterial sample by a sterile inoculating loop and gently make a smear on the glass slide. Now air dry the smear. Next, heat fix the smear of bacteria for 3 to 4 minutes. Now place the slide on a rack of water bath and flood it the smear with malachite green dye and keep it in the steam for 4 to 5 minutes. After that, take off the slide gently from water bath and allow it to be cooled. Next, rinse with distilled water to remove the excess dye. Now apply counter stain safranin over the smear for 1 minute and again rinsed with distilled water. At the final step, observe the slide with oil immersion through microscope at 100x or if possible view under more than 100x for the good magnification and image. So here it's the microscopic view of endospore staining that's done on under 100x. Last but not the least, the mechanism of action of the stains. Malachite green is a water-soluble dye that is alkaline in nature. When the smear is heat steamed, it softens the outer coat of the endospore and allows the dye to penetrate into it. Malachite green dye weakly binds to the spore wall. That's why if it's washed with water, it's easily washed away, and so the heat steam process is necessary for fixing the dye in the cell. Malachite green and safranin acts nicely on bacteria because of its alkaline nature that is positively charged while the cell is negatively charged. An attraction works between the malachite green dye and the bacterial cell that helps to absorb the dye. While examining under the microscope, the vegetative cell form will appear as pink-red in color of counter stain, while the endospore will appear as green in color. That may exist freely or within the red cells, or that will take up the color of malachite green dye. Thanks for watching. Subscribe for more.